My training is in biochemistry, but I worked for many years in an endocrinology lab at Prince Alfred Hospital, and when I was there I was involved both in research into endocrinology and also into the diagnostic side of endocrinology. So I've had a bit of training sort of across the spectrum of laboratory science and also research. Well, you might think the Growth Research Group is a strange name for a group of cancer researchers, but actually it all started back in the endocrinology lab when we were working on growth factors that cause normal body growth and things that go wrong when children, for example, grow abnormally. So we devised a series of tests that could be used, in fact some of them are still used in hospital laboratories to measure normal and abnormal body growth. And it turns out that those same growth factors that are involved in normal body growth also are among the things that go wrong when cancer cells grow abnormally. So over a number of years, our work more or less evolved from studying normal body growth and cell growth into studying the unregulated or dysregulated growth in cancer cells. But we retained the name Growth Research Group. That was our historical name, and we haven't seen any reason to change it. Well, we have two main focuses in the breast cancer work. The first one is to try and work out new prognostic tests for breast cancer. That is blood or tissue tests which will help to predict the course of the disease, and that would help clinicians in managing patients with breast cancer. The second, which is really the bigger research program, is looking at the mechanisms by which breast cancer cells grow abnormally. And once we've worked out those mechanisms, trying to find new drugs or use existing drugs to block the growth of the cancer cells. So really one side is on the diagnostic side and the other one is on the treatment side of breast cancer. Well, the type of breast cancer that we're particularly interested in is called triple negative breast cancer. So the majority of breast cancers have certain proteins in their cells called receptors, like the estrogen receptor or the progesterone receptor or the HER2 receptor. And these proteins are used as drug targets. That is, there are therapies have been devised which particularly attack these proteins and allow cells that have those proteins to be treated, or cancers to be treated. Now, triple negative breast cancer doesn't have any of these target proteins, so that makes it much harder to treat. And the normal way of treating triple negative breast cancer is by using chemotherapy, surgery and chemotherapy. So, I mean, chemotherapy can be very effective, but these days most cancer treatments are heading towards more targeted or personalised therapies. And so we've been trying to work out a personalised or targeted therapy for triple negative breast cancer. So as I said, triple negative breast cancer doesn't have certain proteins that are traditional breast cancer target proteins, like the estrogen receptor. So our job really was to find other target proteins or biological pathways that we might be able to target rather than those other conventional targets. And this really took us back to our research over many years in growth factors, particularly a protein called IGF-BP3, which stands for insulin-like growth factor binding protein 3, a protein that on the one hand carries small growth factors in the circulation, but on the other hand works inside cancer cells and other cells to regulate their growth and their survival. So it turned out from our research that IGF-BP3, as we call it, is a sort of trigger, if you like, that initiates certain growth stimulating pathways in triple negative breast cancer cells. Now, it's particularly important in this type of breast cancer because this protein, IGF-BP3, is present at very high levels in this type of breast cancer, whereas it's much lower levels in other types of breast cancer. And associated with this is another protein called the EGF, or epidermal growth factor receptor, which is also known to be very highly expressed in triple negative breast cancers, but not in other types of breast cancer. So our work led us to discover that this protein IGF-BP3 actually activates the EGF receptor, and this is known to be a strong driver of cancer cell growth. So if you like, we worked out a pathway or a sequence of biochemical events that happens inside these breast cancer cells, whereby one trigger protein, IGF-BP3, stimulates other proteins inside the cell, which then make the cancer cells grow more rapidly. And by having this information, we've been able to work out key points along the pathway that we might be able to use drugs to block. In fact, we've been able to do that successfully. Well, it turns out that to block the biochemical pathways that we think are important in these types of breast cancer, we have to block biochemical reactions in two separate points. And to do this effectively, we need two different drugs. When we had worked out what drugs we thought would be effective, we tried them separately, and when they weren't fully effective alone, we thought it might make sense to try them in combination. And we were absolutely thrilled with the results there, because it turned out that working together, these drugs were really spectacularly more effective as a combination than either drug used by itself. And so we've now developed, spent several years developing this new approach to triple-negative breast cancer therapy based on a particular combination of drugs. Now, there are different drugs in each of the two categories, if you like. It's not two unique drugs. It's a drug of one type, which is an EGF receptor inhibitor, plus a drug of another type, which is to inhibit another protein called sphingosine kinase. That's also activated by our trigger protein, IGF-BP3. And a combination of drugs that work against these two proteins turns out to be very effective in blocking the growth of triple-negative breast cancer cells. What we'd like to do next, if we can raise the funds to do it, is to test our new drug combination in women with triple-negative breast cancer, because I think we've pretty much done as much preclinical work in the lab as we can do, and everything says that this is likely to be a useful new therapy. Now, to do this, we first have to tread very cautiously. We would have to test the safety of these drugs used in combination on very, very small numbers of women, starting with very, very low doses and slowly escalating under conditions where we were absolutely certain that there were no negative or adverse effects. So this would be the normal way new drugs are tested in patients. And although these aren't actually brand new drugs, their use as a combination will be brand new. So we still have to assume that there could be dangers and tread very, very cautiously until we've proven that they're safe. Once we're able to prove that the drugs are safe at the doses that we think will be active therapeutically, we would then try and get a slightly larger group of patient volunteers who would be prepared to have the drugs tested under conditions where we would hope to see a therapeutic effect. So that might be what's called a phase 2 clinical trial. The toxicity testing would be a phase 1 trial. In a phase 2 trial, we would test the combination of drugs at a safe dose in women with triple-negative breast cancer, and then set the endpoints like tumour shrinkage or extending patient well-being by various measures. And if we were fortunate enough that we were able to get through the phase 2 trial and showed real activity of our drug combination, then we could open the door to a large phase 3 trial where this drug combination is tested in wider populations of patients. So this is a long process. We're talking about at least a year for phase 1 or toxicity and safety testing, probably a couple of years for phase 2, testing in small populations, and then a few more years to test under phase 3 conditions in wider populations. So to take drugs, even drugs that are already in clinical use, into the clinic as a new therapy is a very extended process, and unfortunately a very expensive one too. With absolutely brand new drugs, this can cost millions of dollars, and even with drugs that are already approved for use for different purposes, we would expect this to cost many hundreds of thousands to even millions of dollars. If these clinical trials turn out to be successful, and I guess I'd have to point out that many drugs that look really promising in the laboratory never make it to the clinic, because for one reason or another, when they're tested in large patient populations, they turn out not to be as effective as they had previously appeared. But if it turns out that this drug combination is effective in small and then in large populations of women with triple-negative breast cancer, it could have a real impact on the future treatment of this disease. So one thing that I didn't mention is that as well as being an effective treatment by themselves as a drug combination, we've also found that the combination of drugs, even used at lower doses, helps cancers to respond more effectively to commonly used chemotherapies. So there are two possible ways this drug combination could be used. Either by itself, completely avoiding the need for chemotherapy, that would be a wonderful outcome, but perhaps less likely. Or possibly more likely, the drug combination in lower doses, which might be safer doses, used then to allow chemotherapy to be used at a lower and therefore safer dose than it would have previously been used. So it would be the new drug combination plus low-dose chemotherapy. And this wouldn't be unusual in cancer therapy to use these kinds of combinations. It's quite common these days to combine several drugs in this sort of way. But the benefit to patients is that all of the drugs could be used at lower, and that means more safe doses. And hopefully we would see the most effect possibly in that sort of combination. Well, as you can tell from my age, I've had a fairly long career in research. As I said, we had a lot of success in learning how normal cells and normal humans grow over the years, but I think the cancer research in many ways is more exciting. This project that I've talked about, in fact, is one of the highlights of my research career, I would say. Because a lot of our work has been looking at basic mechanisms, but mechanisms where it wasn't clear immediately how these would translate to the clinic. Although, as I mentioned, some of our diagnostic tests are used clinically, so that's very important. Why I think this project is so important is that it has a real opportunity in using currently available drugs to treat a type of breast cancer which has really been quite intractable up to now, and for which the patient outcomes are much less promising than for other types of breast cancer. The reality is in 2016 that breast cancer, most types of breast cancer, are quite treatable, and a diagnosis of breast cancer for many, many women is by no means as serious as it used to be, because they can be completely treated with very long survival. That's not true in general for many of the women diagnosed with triple-negative breast cancer. So if we could make a breakthrough in treating this form of the disease, that would really be, from a research point of view, a highlight, and of course from a patient point of view, a very important outcome as well.